Well, thank you, Christine. It's really a great pleasure to be here. And I do apologize. I wanted to stay another night in Vichy. That was the plan. But there's a strike tomorrow that is blocking all this road. So I want to get back. I need to go back to reality in the U.S. So just how do I start? Oh, this is before. Where is? This is interesting, but we'll get there. Just a second. So everyone is scratching, but I'm trying to see where is the full. Is your guy? Can you put the full screen? Oh, okay, so now it works. So I'll discuss something that all of you are exposed in your lifetime, is itch, and it's a very important component of our daily life because all of us itch at some time. And acute itch, I think, is not an issue in terms of because it is self-limited. You get an insect bite, or some kind of irritation, but the main focus is the work I do is chronic itch. By the way, I don't know if you just saw that the suffix of my name is itch, so I was born to deal with it, and it's basically chronic itch is defined as itch that lasts more than 6 weeks, and I think it is important to know that all 2- and 4-legged animals itch and scratch, and also our leaders, and I don't want to go into politics, but you know what we are facing in the U.S. So we scratch a lot. But chronic itch is a disease state, and that's what really I think is important to understand, and there's a lot of involvement of the skin, and it encompasses a lot of conditions. And I know most of you are researchers in the cosmetic industry, and a lot of the compounds are, causing itch, and I think is of major interest to know which ones are helpful to also the healthy being of our patients. But there are a lot of causes of chronic itch. Most of them are dermatological, like eczema, psoriasis, hives, urticaria, but there are also systemic conditions. There's also neuropathic. There's also a lot of issues of psych involved. Stress is a major issue. But what I'll focus today in this brief lecture is what causes itch. Basically, there's an interaction here between the skin, keratinocytes, neural system, and the immune system. Dr. Kabashiyama, I think yesterday I missed his talk, but I'm quite sure that he mentioned a bit the crosstalk between nerves and the immune system. And I focus mainly as a narrow aspect of it. But the first understanding is that itch is transferred by C nerve fibers. The majority of itch comes from C nerve fibers, and these C nerve fibers extend up to the stratum glandulosum. So they're very in very close proximity to environmental changes, and they can detect changes such as temperature changes, changes also in humidity, and they can transfer itch. There are 2 major pathways of itch, and that's important to understand because the classical pathway that until a decade ago was considered the major was histaminergic. And it's a big industry, antihistamines you can find in every pharmacy, but basically in chronic itch, their effect is very limited. And there's a non-histaminergic that is mediated by a pathway that is activated by serine proteases, tryptase, and activates a proteinase activating receptor 2. Just to explain, the pathway extends to a pathway that in anatomy we know is very similar to the pathway in pain, the lateral spinothalamic tract. and it goes to areas in the spinal and go up to the brain. I did a lot of work on brain imaging and itch, but I think that today I won't focus on that because there's enough to discuss in the periphery. But it's important to understand that there is a big distinction between pain and itch. In pain, you could have, you don't need skin, you can have visceral pain. But in itch, you really need the skin and the upper layers of the skin, the stratum corneum and dermal epidermal junction, epidermis. Now, if you remove all of these structures and go to the deep part of the skin, you can get pain, but you won't get itch. And I think that it's important to understand that there's multiple players. So there's no one receptor that will target all the types of itch. And the main two groups are G-coupled protein receptors, such as receptors for PAR2, which is a G-coupled protein receptor, receptors for opioids. There's an imbalance between mu and kappa opioids. There are receptors for substance P that is called neurokinin 1 involved in itch. The list goes on and on. What is interesting is the mast-related proteins. These are nerves that are more itch selective. They don't transfer pain, but they transfer the MRGPR, and MRGPRX2 is important, especially also with allergic reaction. And one of the issues that my group is very much involved is not just looking at T cells. This morning there was a lot of discussion about immune cells, but T cells are just one type of cell involved in itch. Mast cells, clearly you know, but also neutrophils and basophils can transfer itch. Another type of receptors that are very much involved in itch are the ion channels. The TRP, transient receptor potential ion channels, some of them you know, capsaicin has a receptor called TRPV1. This is more involved in acute itch, and TRPA1, which is a receptor for another food, and I have to tell you the food in France is outstanding, but you don't have so much spicy like wasabi. So wasabi is a receptor for anicarin TRPA1, but it's extremely important actually in itch of different chronicities, in particular in atopic eczema. This figure is very comprehensive, but I would like to mention that the types of itch there are chemical that are mediated in the spinal cord by a receptor on molecular level of gastrin-related peptide receptor. There's also now mechanical itch. So when we wear clothing, sometimes it can cause itching, and this also is kind of inhibited by a receptor, NPY, neuropeptide Y. There's also interactions with the brain, as I mentioned, that I won't discuss that, but know that there is also a top-down effect. So it could be an inhibitory effect from the brain on itching. There are inhibitory interneurons in the spinal cord, but there's also activation of itch. So meaning that you could aggravate your itch. And one of the common conditions are stress. So my Japanese colleagues and in my group, there's a large Japanese representation, is working extensively on what really triggers emotionally itch, and it's an extremely important aspect. One of the other issues, and this has been a debate for a long time, is it was actually coming from the Japan kind of school of thought that itch is related to more nerve fibers in the epidermis. And my group for years was arguing that actually the opposite happens. It's not the number of nerve fibers in the epidermis that is important, it's what the quality they convey. And this study from a former fellow of mine who we collaborated Dr. Tay from Singapore shows with a 3D imaging that this has been recently accepted to JID but you could see how significant reduction in the epidermis of the number of nerve fibers in atopic eczema. This is common in many cases of chronic itch, so it's not really the number. We think that there are some inhibitory pain fibers that are reduced, and therefore there's more itch representation. My team has been for a long time interested in some of the genetic aspects of chronic itch, and today there was already discussions about a lot of differentially expressed itchy genes associated with different conditions, and also in itch, what we were looking is actually comparing non-itchy skin of atopics versus psoriatics versus healthy in humans, and we also worked in primates, but the idea was to really look at correlation of itch to these genes, and it's a long kind of, there's a lot of involvement of many genetic factors involved in receptors that are involved in itch, but there are unique flavors for each disease. So atopic dermatitis has specific cytokines that are involved in itch, such as IL-4 and IL-13, but there are a lot of commonalities between atopic eczema and psoriasis. These are the major two conditions that are extremely itchy, and just to point out is that tryptase, the serine protease, is also elevated in both diseases, substance P in both of them, as well as its receptor. Important to mention is that there are itchy cytokine, and I'll mention that briefly later on, interleukin-31, and this comes from actually work from my colleague Bernard Homey, and then Japanese researchers that looked at IL-31, but also other cytokines like IL-17 that has been highly involved in psoriasis, but has also a role in atopic eczema itch. There are also ion channels, as I mentioned, have a significant role, and there's also sodium channels that are known in neuropathy. I know that some researchers here are working on neuropathy, so it's important to know that NAV 1.7 and 1.9 are highly expressed in chronic itch. We did also immunohistochemistry showing that tryptase is significantly, as I mentioned, not just on the RNA-seq level, but also on immunohistochemistry on the translational aspect of it, is that in atopic dermatitis as well as in psoriasis, there's overexpression, while only part 2, which is that receptor, is overexpressed in atopics. Again, also in psoriasis and atopics, in both conditions, substance P is an important factor also in neuropathic pain or in pain, but its receptor, neurokinin-1, is overexpressed in both conditions. Now, the neuroimmune interactions are really a new concept. Only in the last 5 years we understood that actually nerves talk to immune cytokines, and clearly we have in the DRG, in the dorsal root ganglion of the nerve, receptors for cytokines, and among the most important, as I mentioned, IL-31, but also IL-4. This is the pathway that works on the drug Dupixent, the French drug. IL-13 also activates this pathway, and also the pathway of STATs. And also, I would mention that, as you could see, that there is a lot of interactions between keratinocytes and these immune cells and the nerves. And as I mentioned, interleukin-31 has been coined the cytokine, itchy cytokine, and in my group we've looked at this cytokine in different diseases. One of the diseases that I'm quite sure is very highly associated with this cytokine in itch part of it is cutaneous T-cell lymphoma. This is a Th2 cytokine, highly expressed in atopic eczema, but in CTCL is highly expressed, both its receptor as well as its ligand IL-31, as you could see here in the epidermis and dermal epidermal junction. Another aspect that needs to be addressed when we talk about itch and when its treatment is the itchy skin, meaning the sensitivity of the skin. And this is something extremely common, and it's called allonesis. What is allonesis? Is that a non-itchy stimuli in patients with chronic itch have a very high sensitivity to transfer itch. So this explains a lot of times why patients who have chronic itch say that they put clothing on, and it doesn't have to be the wool fiber that we always are talking about. It could be other clothing when they change clothing or there are changes in temperature, that usually they don't induce itch by itself, but it does in these patients. And this could be a peripheral mechanism, such as nerve growth factor and its receptor TRK activation, but it could be, and also prostanoids, but it could be also on a central level, and part of it is neurokinin 1, the receptor for substance P. We have also shown that on the brain also, there are changes in the brain that are associated with neural sensitization, especially in the anterior cingulate cortex. So kind of summarizing, this is a very important aspect of treatment of chronic itch because you have this sensitivity of the nerve. So in order to reduce it, you have to dampen that sensitivity. I thought that it would be nice to mention a study that just was published last week in JID, where we collaborated with another Frenchman, Olivier Josidov, on scabies. As you may, those of you who are dermatologists know, he's a world expert in scabies, and he was asking us whether we could examine the neural aspects of itch in scabies. And why is it important? Because for years we've been giving antihistamine to these patients. I was never impressed that it helps the itch. Scabies is the most itchy mite dermatosis and common. It's dreadful for dermatology residents and faculty because it's so contagious. We talk about 100 million patients a year suffering from it. So the question was really what is the neural aspect and mediators involved? And what we've shown was that actually histamine has no expression or overexpression in comparison to healthy. In both a pig model of mite induction and also in human biopsies. And what was highly activated, not surprising, as I told you, that chronic itch in the majority of cases is activated in the proteases in part 2, but this was the first study to show how significant part 2 is highly elevated in this population of itchy subjects. Not just part 2, the TRPA1 as well as TRPV1 were highly expressed in this group. So again, sometimes it's worth to re-examine a lot of our thought processes. For sure, I think that this is a further evidence that antihistamines don't work for this population. So I am a clinician scientist, so clearly I want the developments from the basic lab going into the bedside. So my common goal is to develop better treatments for the patients, and I think that the message I have here is clear, is that antihistamine in the majority of cases of chronic itch don't work. So what type of treatments do we work with and publish? We've used ion channels, as I mentioned. Ion channels have a significant effect on itch transmission, so we have used a topical preparation of ion channels inhibitors, such as using ketamine, which is an NMDA antagonist, with lidocaine and amitriptyline in a lipoderm base, and we've shown that this reduces itch. This is maybe the most robust topical that we have available. Another treatment that actually some French dermatologists also used was botulinum toxin. We've actually done a double-blind study that has not been yet published, but showing that it has a long-term effect, again, targeting the nerves and the ion channel. We've also been involved in a development of a tropical TRIC-A, which is the receptor for NGF, and this worked only in psoriasis, and not in atopic eczema. So not every drug that will be developed would be fit for all types of itch. And another topical PDE4 inhibitor, I think, is also available in Europe, that is a phosphodiesterase 4 inhibitors, such as crisaborole. They have itch reduction mainly in eczema and some parts of psoriasis. I think that this figure is very comprehensive in the amount of treatments that are available now, and some of it is developments of a French company. I'm not promoting Sanofi or others, but and I have a conflict of interest that I work with all of these companies, but Sanofi actually opened our field, and I have to acknowledge that because now there's so many new drugs that are targeting the immune system, and as I mentioned, the cytokines are not just involved in the itch of eczema, but in other diseases, in psoriasis, and it won't be surprising that other conditions that itch has an inflammatory component could be reduced with it. So clearly, nemolizumab is another drug that is an IL-31 antagonist, and Dr. Kabashiyama had beautiful papers in New England and I think Lancet showing, and Jackie in Journal of American College of Allergy and Immunology showing that this drug has a significant importance in itch reduction, maybe even stronger than dupilumab that works in atopic eczema. But the list goes on and on. As I mentioned, IL-17 antibodies such as secukinumab and ixekizumab have antipyretic effect. And one of the hot topics now are the Janus kinase inhibitors like tofacitinib and others that are targeting a broad T-cell antagonism, and they have an antipyretic effect. So stay tuned because this field is growing. And I think that we covered that, but another aspect of treatment is reduction of that neurosensitization that I mentioned. So using GABAergic drugs reduces that nerve sensitivity, and that's why these drugs are used in my armamentarium. And we've also been using drugs that are antidepressant from the SNRI, selective neuroepinephrine reuptake inhibitor, such as mirtazapine. It has an effect also on nerve fibers and anti-nociceptive effect, and the combo of using it with gabapentin and pregabalin reduces itch. And I mentioned to you also that there is an imbalance sometimes in chronic itch between opioids, meaning opioids, as you know, are anti-pain, but mu opioids, but they actually cause as a side effect itch. And colleagues in Japan more than 2 decades ago nicely showed that in patients with chronic itch of uremic pruritus, there's an imbalance of mu overexpression in the skin as well as in the spinal cord and kappa receptor, which is downregulated. That led to the development of a drug in the Japanese market called nalforafin. Unfortunately, this drug did not make it. It doesn't have a very strong antipyretic effect, but other drugs have been in development, such as nalbuphine and asimodulin. So most probably these drugs will make it to the market in the US because we don't have yet these drugs in only in clinical trials. More than a decade ago, I found that there's actually a drug called butorphanol. I assume that this drug is also available in France, and it is a kappa opioid agonist and a mu antagonist. Another hot topic in treatment, as I mentioned, nerokinin 1 is overexpressed in chronic itch, at least in atopic and psoriasis, and most probably in also other conditions such as prurigo nodularis. And again, there's a lot of new developments of drugs that are working on this receptor. there is a drug in the market called the Prepitent. It's very expensive and it's very difficult to administer because it has too many drug-to-drug interactions. But there are newer drugs like Serlepitant that we were involved in clinical trials showing that it has a significant antipyretic effect. So I think that the beauty of it is that there's so many new targets, a lot more interest. If you would be in a meeting 10 years ago, there weren't any of these drugs in development, and this field is really hot. So just to kind of summarize, I think that there's a lot of excitement in this field of itch. We need to target the narrow immune interface. I didn't mention that the MRG and especially MRGX receptors seem to be a very valid target. And again, reducing neural hypersensitivity with drugs that target the neural system would most probably reduce the itch intensity. And for sure, we don't want drugs that have too many side effects for our patients. So the work that we've been doing for the last 2 decades is a lot of collaborations with different groups, including my French colleague Olivier Roussydov and Charlotte Bernigat. But a lot of the work is with different groups, and this is my research team, and I would be happy to, in all my funding resources, I'd be happy to address any questions that you have. So thank you again for your kind invitation.
Thank you. Is there some questions? Frédéric?
Thank you very much for your presentation. I have 2 questions. The first is about psoriasis, because we learn to medical students that normally it's not an itchy dermatosis, but I agree that in around 40% of patients they have itch. How can you explain the difference, patient with itch and other without with the same disease?
Okay, so it's an excellent question, and I agree with you. I mean, I was standing 20 years ago, I was practicing in Singapore at the time, and just to let you know, a world expert in psoriasis at the time came to Singapore, and I had the study showing that around 80% of patients with extensive psoriasis have itch, and he said, I never saw a psoriatic patient with itch. And then my colleagues in Singapore said, Gil, aren't you ashamed to publish that? And I said, I'm standing by my words. I mean, this is our patients. And it's interesting that just now, almost 20 years later, every drug company is looking at itch and psoriasis. So it's so a hot topic. So part of it is our blame. We didn't ask these questions. So when we don't ask, the patients don't tell. So it's a major problem. Now, in regard to your question, so clearly there are other factors involved in that. So it's not related to the PASI score, and the severity is only one factor. There are narrow immune interactions there. There's clearly interesting differences between it and atopic eczema. For example, I could say that we found that the cold receptor is actually activating itch. So this is something interesting because we found that a lot of times we tell our patients to cool your skin when you have itch. And apparently in psoriasis, it aggravates itch. So, I mean, we're learning in the process. Neurokinin-1 receptor has a role. There'll be soon results of a large study done with that. The NGF receptor that doesn't work for some reason in atopic eczema is very effective in psoriasis. And a lot of it is, again, neuropeptides, growth factors that are involved, cytokines such as IL-17, other cytokines. TNF is less important, but has some role. So yes, good question. There are aggravating factors. So stress, if you recall, is a major factor in psoriasis. So I think with patients who have more stress or have more kind of prevalence of itch, there's other interesting aspects of it. The scalp is the most itchiest of psoriatic areas. So you should ask in atopic, rarely we see atopics that itch so much in their scalp. So there are some unique features that we'll need to explore further, but it's an excellent question.
And my second question is about scabies. So you mentioned the work you are doing with Olivier Josipov and Charlotte Bernigo. And about this disease, I have a question, because normally the parasitic load is very low. It's around 20 parasite on the complete skin. And in fact, the itch is very severe. So it's something it's not obvious for me.
Yeah, excellent. So I think that the parasite induces an immune response. I didn't mention, this is for our next publication, but the cytokine IL-31 is really highly expressed in macrophages and in these patients. So that's one of it, but it activates a non-histaminergic pathway, and clearly the part 2 downstream, there's a cascade of immune cells involved. As I mentioned, IL-31, but there's more. Stay tuned, because we're really working on these issues more carefully, and we have also some specific proteases that the mite induces itch, so it induces a cascade of activity. And I agree with you, you don't have to have, you can have one mite that induces all the trouble. So clearly it's not just the amount of mites causing that.
So if you think it's related to immunity, do you think it's an explanation for association of bullous pemphigoid and scabies?
I'm sorry, can you repeat that?
Do you think it could be an explanation, because you tell that it induces an immune response? Sometimes you have association of bullous pemphigoid and scabies could be related?
Bullous pemphigoid, that's interesting. I think that there are, bullous pemphigoid is another interesting area, which is very itchy, of association. Is it the same mechanism? I'm not 100% sure, but clearly the immune system plays a role in both conditions. So yeah, and post-scabetic syndrome is, is something that is very common in our patients. So when with ivermectin, you destroy the mite, but the patient continues to have for 3 or 4 weeks still itch. So clearly it's not just the mite inducing.
Lionel?
Okay, so I have,
Yes.
Thank you, Gil, for your talk, really nice. I have one question and one comment. My question is about, so I totally agree with you that so it's not the number of nerve fibers that could be relevant, but probably the level of stimulation of each of them. Did you check the differential potentials that you should apply in this kind of condition on the nerve fiber to stimulate endogenously, stimulate versus control? Do you have because I'm wondering since a long time that there is a small difference of level of potential that the body should apply to the nerve to stimulate. In the case of itchy condition, do you think that the differential potential is really small that could explain that the stimulation could be more frequent or something like that?
It's an excellent question. We are actually did not examine in psoriasis, but I do believe that you're perfectly on target. In atopic eczema, we've shown and published last year in pain that atopics have that neurosensitization in the periphery significantly. So they have a neurosensitization. It's not just for itch, it's also for pain. Another area that is now becoming hot topic in this field. So, there's a mechanical and chemical neurosensitivity in these patients. So very, someone? I'm sorry? Someone had a comment?
No,
Or-
no.
oh, I'm sorry. So basically, I think this would be our next step. We applied for a grant to look in psoriasis. Won't surprise me that it's a very similar mechanism.
Interest. And my second is more comment. So you mentioned neurokinin 1 antagonism and NK1 antagonism.
Je vous rappelle.
Sorry? You hear me?
No, 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 no. Continue.
Okay.
Don't stop, Lionel.
It's so. I think
I
that you are.
found the echo, but
Yes, yes. They think probably that you are the last candidate. I should confirm that Gil is not the last candidate, so it's not to be challenged. So just to go back to my comment, so you mentioned NK1 antagonism, classical approach to receptor binding and so many things like that. As this symposium is organized in Vichy, and we could find many sports spring motor in terms of not antagonism against NK1, but on functional decrease of release. Do you check this point? And as we could find in Europe with Vichy, with Aven, and many things, and many sports spring water like that. So in US, did you check this kind of spring
water,
sport
yeah. Excellent
check?
question. So in reality, I do believe something. For years, and I'm not trying to Aven water, I remember recommending using it for soothing effect. I'm quite sure that there is something in addition. I didn't check that. Unfortunately, I didn't get funding for that, but if you'll be able to and interested that L'Oreal will, trust me, we will check that and we'll get some results. I won't be surprised that there is some soothing effect of these minerals, and because they may work on some of these sodium channels, or the issue is it's difficult to do it. So I mean, to design it is not just to do usual tap water versus the mineral water and see what happens. We have developed a model that you could actually test in L'Oreal with a cow hage that companies do use, and that model is very mimicking to the chronic itch of a lot of diseases. So it's kind of an easy study to do, if you're interested. Yes?
Yes.
Please. Did you look at the expression of the other receptor of NGF, the P75?
Yes.
That could explain why sometimes you don't have the effect of your inhibitor.
In this, we didn't see P75, but in another disease, we looked at it in lichen amyloidosis. There, also, it makes sense to look on that. It's also been associated with some apoptotic, and at the time we thought that, one of the explanations, unfortunately we did not show that, but we thought that, in aging, this is another issue that I think the cosmetic industry is in. The older we get, I have bad news for you. The older you get, there's less pain, that's maybe the good news, but more itch. And part of it was we thought that there is an apoptotic effect that is related to P75, but we weren't able to show that. So it's a good point.
So we have to stop
Yes,
the discussion. No. The last one.
Thank you very much for your talk. In your last slide, you mentioned to reduce neuron hypersensitivity, but you show in atopical dermatitis atopic dermatitis, they are less neurons and the skin. Do you explain more, please?
So it's an excellent question, and the idea is that quantity is not equal to quality and intensity. So and one of the explanations you have to understand is the innervation territory of of itch is a lot bigger than in pain. So it could be that you don't need so much numbers of nerves to get that sensitivity. You need some of the receptors involved, and NGF is a sprouting, but we don't show sprouting, but there is a possibility that there is mediators there that intensify the itch perception. So numbers are not everything in life. Does that answer you? It's how the receptors and what the mechanisms of intensification that is not related just to quantity. And I said that quantity, this is more a hypothesis because we didn't show, the older you get, there's less actually epidermal nerve fibers and you have more itch. So again, that sensitivity to itch could be peripherally, but if I showed you that in the brain there is also higher activation in areas that are involved in sensitization. One of the areas in here is cingulate cortex. But again, the cortical area in the brain is reduced in aging. But again, it could be a sensitization of the itch process there.
Okay.
Well,
Thank you so
again.
much, Gil. Thank you.